Hello everyone, my name is Manon Poignet and I'm a PhD student at Charles University in Prague. And today I will talk about some speciation process and more specifically about post-mating prezygotic barrier into nightingale species. First of all, we know that one of the main goals in evolutionary studies is to understand how new species arise. And we know that different type of speciation scenario can occur where species like population of one species will be split and these two populations will diverge to create two new species. But then it's possible that these two species came again in new contact, so-called secondary contact. And if the reproductive isolation are not complete, then some hybrid can occur. Then, in that case, in this secondary contact zone, some new mechanism of isolation can be produced and settled. And these mechanisms are very species specific and not the same one will occur in different species and not in the same way. This mechanism of isolation will be divided in two categories. The prezygotic isolation, subdivided in premating isolation, composed of habitat, temporal, or behavioral isolation, or some post-mating isolation like mechanical or gametic isolation. And the second main group is the post-zygotic isolation after the creation of the zygote, and this will be mainly with the hybrid, and it will be about viability or fertility in the hybrid. But today I will talk mostly about post-mating isolation and more specifically about gametic isolation. And we know that these post-mating prezygotic barriers are mostly controlled by post-copulatory sexual selection, and they are mainly caused by some female cryptic choice or male competition to fertilize the female. And these two main things are driven by ejaculate female or ejaculate egg interaction. And this interaction, if they are really species specific, they can reduce the interspecific fertilization. And we know from all of this that the sperm cells evolve very fast and he present a very high interspecific diversity of morphology within species and between species. So it's a good candidate for a potential barrier for heterospecific crosses. So in our study, we were focused on the common nightingale who live in the southwestern part of Europe and the thrush nightingale who live in the northeastern part of Europe. And both species diverged around 1.8 million years ago, and then they come in during Holocene in secondary contact in the middle of Europe. And here in St. Patrick, where both species occur, we know that 3 to 5% of the population is composed of hybrids. So that means that the reproductive barriers are still incomplete. So we already searched for different type of isolation mechanism that can already be settled between the two species. And it has been found that some premating isolation occur with some partial habitat segregation, but also some post-zygotic isolation because following Aldan's rule, the sterile F1 hybrid female, the F1 hybrid female are sterile, whereas the male hybrid are fertile. And the last one is some post-mating prezygotic isolation signal with the divergence in sperm size. So more specifically about the last one, Albrecht and al. In 2019 found that the common nightingale has a higher, longer sperm compared to the thrush nightingale, and this length was mainly explained by the midpiece. So here is an example of passerine sperm, and you have the head and the flagellum. And the flagellum is divided in midpiece, where you have the mitochondria surrounded the flagellum and the tails, which is like without this mitochondria. So mainly here, the midpiece was different between the two species. And at the head level, it has been found that between population of allopatry and sympatry, it seems that there is some differences, and this can suggest some reinforcement signal. So it's a very good candidate for potential mechanism of isolation of species. So to go further, we did some thin scale analysis in the morphology of sperm of both species, some motility experiment and proteomic analysis. So both species were sampled in Poland, here in sympatry, where both species co-occur, and in allopatry, where only one species is present. So first, we did some morphology analysis with some confocal picture to have small details in the head mainly. So we apply different measurements here on the head, where you have in blue the nucleus composed only of chromatin, and the second part of the head, which is in green here and is the acrosome. And here again, so the head and then the midpiece, so the flagellum with the mitochondria and the tail without the mitochondria. And with some statistical analysis, we found that the acrosome was longer in the thrush nightingale compared to the common nightingale, but the nucleus was not significant. And that was expected because the genome size of birds is very conserved. And we found also that the thrush nightingale had a wider acrosome compared to the common nightingale. For the flagellum, we confirmed that the mitochondria, the midpiece, was longer for the common nightingale compared to the thrush nightingale, and we found no differences in the tail. So to conclude on that part, we found a longer midpiece for common nightingale and a longer and larger acrosome for thrush nightingale. And also, we did not find any differences in the region for all these parameters, so we did not confirm the reinforcement signal for the head. But with this, we can hypothesize that the common nightingale will swim faster because he will produce more energy via the mitochondria, and also he will have less drag due to the large head. To do this, we did some motility experiment where we sampled the ejaculate of both species, and we record the velocity in neutral environment, here some medium, as well as in the common nightingale female fluid to see if there are some differences in heterospecific or conspecific treatment. And for the second one, we used the PBS as a control. As a result, we found that in this neutral environment, there were no significant difference between the two species in the velocity. But what it was surprising is that in the female fluid, for conspecific or heterospecific treatment, we did not find any differences in the velocity again, as well as in PBS. But what we found is that the protein composition of the female common nightingale female fluid reduced the velocity of the sperm compared to PBS, but it reduced in the same way, that were no differences between the two species. And finally, we did some proteomic analysis of the fluid and the sperm of both species. And we found that in the fluid of female, the common nightingale fluid show higher number of unique protein compared to the thrush nightingales, meaning that there are some differences between the two fluids in the composition. And also in sperm, we also found that in the common nightingale, there were like more unique protein, but very in a few number. So it seems that the fluid is more divergent between the two species than the sperm. But that highlights the fact that if we reproduce the experiment of motility, but using the thrush nightingale female fluid, maybe the result will be completely different. but the fact that it was very difficult to catch some female of thrush nightingale in the field, this experiment was not possible. So to conclude on this analysis, we found a longer midpiece and shorter acrosome for the common nightingale and more unique protein in the female fluid, whereas no significant differences were found in the sperm velocity between the two species and in each treatment. So we can think now that maybe the production of energy is not how we are thinking, and maybe there is a different way of utilization or maybe a way of production. Maybe, for example, maybe the head can produce some energy via glycolysis, and that can help the thrush nightingale who has a shorter midpiece to have the same production of energy than the common nightingale. Or maybe that this barrier doesn't occur during this journey of sperm inside the female tract, but in another checkpoint, for example, in the fertilization point where the head is very important, or maybe during the sperm storage tubule. So I would like to thank all the collaborators for this work to help me with the sampling as well as the analysis. And I would like to thank you all for listening.